The overall goal of the following experiment is to introduce glutamate or hypoxia stress as a potential model to test the effectiveness of neuroprotective agents. This is achieved by first obtaining freshly enucleated bovine eyes and performing a quick but still atraumatic explantation of the retina. Next, after superfusing the retinas with a special nutrient solution, electrodes are used to record a series of stable baseline electroretinograms or ERGs. Then, glutamate is added to the nutrient solution, or hypoxia is induced by adding 100% nitrogen instead of oxygen. Finally, an ERG is recorded in order to see the effect of glutamate stress or hypoxia on the amplitudes of A and B waves. The results show a change in retinal function when exposed to hypoxia or glutamate stress through ERG recordings.
The main advantage of this technique over existing methods like cell culture or in vivo ERG recordings is that functional testing can be performed in contrast to cell cultures without having the variability exerted by electrode placement or general anesthesia compared to in vivo experiments.
This method can help answer key questions in the pharmacological field, such as testing neuroprotective agents.
The implications of this technique extends to therapy or diagnosis of acute or chronic diseases, because possible neuroprotective agents can now be tested with this stress model.
Though this method can provide insight into neuroprotection in the ophthalmological field, it can also be applied to other medical areas such as neurodegenerative diseases.
Generally, individuals new to this method will struggle because of the preparation technique.
The model itself was developed by Professor Schnichels in Cologne